Okay, welcome everyone to lecture 11 we're going to talk today mostly about protein folds, stability and thermodynamics of structural transition, focused on folding, but not only. What I thought we would do, so last time we talked, we were talking about bioinformatics, right? This notion of predicting structure and then function from sequences. Today we're doing a little bit the opposite. We're going to see which structures allow which sequences. It's going to be the reverse game. So I thought we could try to see if you guys had questions about last time. If you don't have questions, and also, by the way, if you do, I think during the first part of the lecture, I'll do a lot of, as we go through the slides, a lot of like questions asking you to answer. And I think it's a good, we're coming to the end of the class, right? So everything should kind of fit together. So I'll try to see if you remember some notions from the first part that Eric lectured on, and Burke, and then me also. And you can gauge a little bit how you're doing with respect to the material. The type of questions I might ask can be a bit related to what comes in the exam. So not the exact questions, but kind of the type of things. Do you guys have spontaneous questions about anything, but more specifically bioinformatics? I'm sorry, I only uploaded the lecture video today, so I guess even if you wanted to, you couldn't really review it, but you had the slides. Are we good on the central theorem that says that we have sequence, structure, function via dynamics? Yes, that we can do experiments and simulations to look at how dynamics support function. This is all fine. Your hand in went well? Okay. Welcome, hi. Okay, so then I guess we dive in, and please answer my questions. Don't let me be alone here. I'm super jet lagged. I was in California up to yesterday, so we'll see how this goes. Help me out. Okay, so like I said, the main game today is going to be about fitting sequences into structures, but these are the points that we're going to be traversing. We're going to talk about stability. We're going to talk about how evolution supports structure, so kind of linking really back to bioinformatics. We're going to talk about protein size and do proteins arrange in domains, or do they just keep growing, adding kind of secondary structures to them? And we'll do a little bit of math today and see how the Boltzmann statistics support folds, or maybe it's not Boltzmann statistics, and why that is. So for those of you who like equations, you're going to see equations in this lecture a little bit. For those of you who don't, you're also going to see equations. And we'll talk about specific residues, so we'll come back to, what is a serine versus an alanine, if I have a hydrophilic versus a hydrophobic residue, what comes, and so on. And then towards the end, we'll talk about transition between states, and we'll discuss which types of experiments allow to evaluate if folding is a all or none process, or if it occurs gradually. Okay. Do you guys remember, I think from bioinformatics, how many genes we have as humans, roughly? Like order of magnitude. 100, 1,000, 10,000, 100,000, a million? 10,000. Yeah, good. So we said 20 to 25,000. Remember, the number of base pairs in DNAs was really orders of magnitudes larger. I actually don't remember, but it was of the order of millions. And then we said that many of these base pairs were not involved in defining genes that would then be translated into proteins. So there's a famous quote that says that there's about 1,000 folds for the molecular biologist. This means that even though there are 20 to 25,000 proteins that are important for human biology, there are only 1,000 folds that are found in nature. So nowadays, this is a bit old nowadays, it's more 1,500, but still we're about 10 to 20-fold smaller. Why? What does that mean? Why do most sequences seem to fit in a small number of folds? That's the central question. But basically, this means that there are going to be 10 to 20 sequences that are completely different, but that fold in a very similar way. Yeah? So maybe that's a little surprising. We might imagine, that that for each sequence you have a different fold, and this is really not the case. So today we talk about how physics will put constraints on this and why that is. Back in the 70s, we thought that each protein was a unique fold. And in fact, it was very surprising because the first two structures that were solved were hemoglobin and myoglobin. And they turned out to have very similar folds. So the first two ever. So that really surprised people. Then maybe we started thinking maybe all proteins look the same. So it turns out, no, there's 1,000 kind of different folds, but quite surprising. In fact, this is kind of apparently a principle that holds in many cases. 20% of folds account for 80% of proteins. So 80% of these 20,000 proteins, they fold into only 20% of all the folds that can be observed in nature. How about DNA? This you guys know. How does DNA fold? I mean, I think like your little cousin knows this. Double helix, right? There's only one fold for DNA. And why would there, and DNA we saw are made of 4 different types of base pairs, right? A to T paired together and C to G paired together. But what's the function of DNA? Thank you. Exactly, the only function of DNA is storing information. So it doesn't really need to fold in any specific way compared to proteins that might want to bind a ligand, to have a structural role, and so on. The only role of DNA is to store information, and it has to be unpaired in order to get translated into RNA and then into protein. So it makes sense that there's only a single fold for DNA. Yes?
When you say folding, is there a dynamic of folding? Because in nature we find the DNA already twisted helix.
Yes. So in this lecture, we're talking about thermodynamics. We're talking about end states. We're talking about what happens once it's folded. Remember you had a lecture with Berg Hess where you talked about kinetics of folding, where you talked about barriers and how long it took to get there and so on. So today we're talking about end states. So let's say we can almost ignore, for this purpose of this lecture, the pathways and how long it takes to get there.
When you say fold, it's always going to be the end state.
Yeah, so what I mean by fold is this, right? Like the overall arrangement, the combination of secondary structure elements, beta sheets, alpha helices, and loops into a unit. That's the fold. So this is another type of fold, which is made of 3 helices. So what I mean by fold of DNA is that its structure is a double helix. Good question. Now, RNA is a little weird, I think, because like we said, so we go from DNA to RNA to then protein. So the function of RNA is like an intermediate step, you might think of it that way, in going from DNA to protein. However, this is a little bit of an outdated view. So I think modern biology kind of shows that RNA really can take on many different functions. And this is really an area that is under a lot of exploration right now. And we don't know all of it. But RNA also has a large variety of folds, especially because it can serve to not only store information, but many other things. So why this surprising fact? Why do we go from 20,000 sequences, 25,000 sequences, to a few folds? I have a few notions out here, and let's see if we can review what those are, if you guys remember. So what is the meaning of evolutionary divergence? This is something we covered in bioinformatics. I might not have used exactly this word, but think about what it means. Divergence means we're originating from a same point, right? And going towards different directions. And this is happening through evolution. Do you remember? Common ancestor? Do you want to talk?
Yeah, I don't know, you just mean that you have some random mutations and then by evolution.
Yes.
Some mutations that still remain mutations.
Yeah, that's right. From a common ancestor. Yes, you wanted to say something?
I don't know what has changed. Is it the gene sequence? Is it the structure or the function? Or
Yeah. So here we're talking where this slide is about answering why the protein sequence is similar. So I'm just saying that one of the reasons that a protein sequence could be similar is that they all started from the same, and then through natural selection and random mutation, you introduce small mutations, and then they get selected or not, but the overall fold is preserved. That could be one reason that this happens. Then there's also this notion possible as an explanation for why proteins are similar is functional convergence. Do you guys know what that is? We covered this, I think it's been coming up in the class many times. And I think one example we took was, for example, in membrane proteins. You want to have a fight off again? You don't know?
Yes.
Yeah?
I could just guess that small changes in the structure can still lead to similar functions.
That's right. Isn't that what we just said before, though?
Kind of, but the reason was...
Oh, the other, oh, okay, the previous one, the reason was evolution, whereas now you're saying the reason is function. Is that what you're saying? Mm-hmm. Right, that's true. I think this goes even beyond and kind of says, if you want to perform a special function, and I'm going to go back to the membrane protein case. Let's say you want a protein that sits into a membrane, which is hydrophobic, and that will transport ions, so an ion channel. How are you going to achieve this? You will have to place a hydrophobic exterior something with a hydrophilic interior something into the membrane. And maybe there's not so many ways to do this. Maybe nature can only come up with so many things. That's the meaning behind functional convergence. So again, it's an evolutionary process, but these folds will be selected for, and there will only be a few. And maybe we've seen actually in membrane proteins that a lot of these proteins were just alpha helical, right? There were only a couple beta barrels that were found in bacteria. So it seems to hold. There's not too many ways to arrange an ion channel. What if I tell you how would I structure a protein that is globular to bind a hydrophobic ligand like tryptophan? What's the pressure now onto this type of fold? Globular protein lives in what kind of media? Water, hydrophilic, exactly. So the external the external layer will have to be hydrophilic this time, and it needs to bind a hydrophobic compound. So hydrophobic pocket that will bind the compound. If it's a hydrophilic pocket, slightly different. Now I need hydrophilic, hydrophobic, and hydrophilic inside. Yeah? Simple. So we've covered these two already. And today we're going to talk about this third possibility, which is what are the physical restrictions that can be placed on the protein structures. So it will be in some way related to function, but you'll see we'll take a more ab initio kind of approach where we start from first principles and we derive simple physical models to do this. So what do you think then? Why are proteins similar? Can we pick one of these three? Is everything I said just true, or is one more true, or?
I think the less true is evolutionary dynamics. Less clear at least.
The evolutionary divergence is less clear. Why?
Because the two others are more clear. I mean, the same function should lead to, if you go backward, same.
Is that because of your background that you're not used to thinking in terms of evolution, you think? That it's less clear?
But it seems, I think like if we have a limited number of possible folds,
Mm-hmm.
then you can explain functional convergence.
Mm-hmm.
Because if you have like, if you want to achieve a function through evolution, then you will have a number of possible folds to achieve this function, and then through evolution, then you will have to converge to one of these folds.
Right. So I think the answer will be, it's a combination of these three things, and depending on which specific case, one might play a bigger role than the other one. So some things, some specific functions will be more constrained by the functional aspects, and some of them will be more constrained by the evolution. And many times we don't know. Okay, thank you. That's good. So I told you that the first two structures we ever got were hemoglobin and myoglobin, but then throughout the late 70s, early 80s, going into the 90s, we started having enough structures that it started becoming intriguing to group them into categories. You know how that happens, right? We just have too many things to consider them at once, so we try to cluster them to understand them. And so this is a scheme that you find in the Finkelstein book that shows you a specific classification into different types of architectures. And basically, they are organized in 3 different categories. The red quarter over there are the structures that are made only of alpha helices. And note up there the globin fold. So that's the one that will, if you put 4 together, you'll form hemoglobin, myoglobin. Then this green corner is folds that have only beta sheets. One famous one is this Ig-like because it's involved in immunorecognition, so in antibodies and so on. And then in this big yellow half are all the structures that contain both helices and beta sheets. And you can recognize this jelly roll, trefoil, tin barrel, alpha beta plates, doubly wound, and so on. You guys, I think, are, I hope, now clear on what an alpha helix looks like and what a beta strand looks like, and that beta strands arrange together to form beta sheets. Do you remember why we form preferentially these secondary structures? It's an energetic property, right? We want the folded structure should be more stable than the unfolded structure if we want the protein to fold and to be able to perform its function. Do you remember where the main energetic contribution comes from? Hydrogen bond, that's right. So remember in your alpha helix, you're forming a hydrogen bond between the CO and the NH of the amino acids that are located at position I and I+4, right? In beta strands, you had hydrogen bonds on either side that would form to the strand that would be placed next to it. Yep. So these folds seem to all involve having stable local patterns that contain a lot of hydrogen bonds, either alpha helices or beta sheets, and then they come together to form these specific motifs. So by the 90s, we knew we could classify these proteins that way. And there are specific data banks that do this that you can find online. So this is a little bit linking to the previous lecture on bioinformatics where we talked about where you could find information like organized into specific, organized specifically. So there are 2 big types of databases that describe the fold classifications. The first one is completely automatic, and it's basically an algorithm that sorts through, that takes any structure that gets deposited in the protein data bank. Remember, that was the data bank that contains all the protein structures. And it arranges them hierarchically into four different layers. So first gets classified into classes, and this is what we just saw before. Alpha, helices, beta strands, or alpha and beta mixed together. This forms an architecture which then gets put together into a topology. So these tin barrel sandwich and rolls is what we saw on the previous slide in this diagram. And then there's another layer which has to do with homology. And that's not directly derived from the first three, but it's about finding interesting commonalities between classes of proteins that is related to evolution. Remember, we used this principle to build protein structures in the bioinformatics class. So this database is kind of cool because it's automatic, so it means there's a lot of things with it. And again, because it's automatic, the downside is that it contains but it contains information that is not completely correct just because it was done automatically. This other database is the opposite. It's called SCOP2, and it means structural classification of proteins number 2. And this, to me, is amazing because it's entirely built by hand. So then behind each structure, there's a person that goes and curates it into its own fold. And you might imagine that these two philosophies might be a little bit different, because if you're a biologist and you know things about the biology of proteins, there might be features that you ignore because you know they're artifacts either from the structure determination or maybe a specific fold has an extra helix hanging out there, and this biologist knows that this is not important for function, so they don't consider it and don't put it in there. So that makes a database that is slightly different. Two different philosophies. So we'll take an example now, and we'll talk about whether this classification reflects evolution. So is evolution putting pressure on the fold or vice versa? So the example that's taken in the book we're using for this class is again about hemoglobin. We saw this fold, and the observation is that the affinity, so hemoglobin, the role is to bind oxygen from the blood and then release it to myoglobin so that the muscles can use it, right? So it turns out llama hemoglobin binds oxygen with much higher affinity than the horse hemoglobin. Can you guys think about why that might be? Where do llamas live? Up, so on the high plateaus of the Andes. In this kind of environment, oxygen is rarer, right? So it makes sense that the hemoglobin needs to bind the oxygen with higher evolution because there's going to be less oxygen in the blood. And then there's another example that's given in the book, which is, quite interestingly, the hemoglobin from fetus in the plural, fetuses, is different from adults. And can you guys think of what might be happening in the case of a fetus? So I'm not telling you if it binds oxygen harder or weaker, but what's your guess? I guess it's one. Who was talking? Okay, do you want to explain why you think that is? Yeah. That's right. So it's a little bit of a sad story in which you consider that the fetus is trying to steal the oxygen from the organism that is feeding it. hence the mother. So that's right. So the fetal hemoglobin will bind oxygen better, and so that way it can extract the oxygen from the blood of the mother over the placenta. What is interesting, and is not central to this specific class though, is that the genetic material of the fetus is the same as the genetic material of the parents, or it's a combination of both, right? It's not that it's different. So this means that the DNA is the same. So it's actually the expression of the genetic material into proteins that is different. This means that the genes that are activated in the fetus development for hemoglobin are different from the genes that are then activated in the mother. If you had the gene for fetal hemoglobin that would be activated in the mother, then it would not, the fetus could not survive. So evolution has made sure that oxygen can bind enough, but also not too hard because it needs to be released later. So based on this, I want to ask you the question, if I go from one organism to another, do you think that the proteins will be more advanced, more complex in a higher organism than in a lower organism? So if I compare a bacteria with let's say, a human or a llama, which one is going to have more complex proteins? So I'm not talking about number of genes here. I'm talking about the architecture, the folds, how they're put together. Should we take a vote? Who thinks that the bacteria have more complex proteins? Okay, who thinks that the bacteria have less complex proteins? Do you guys want to defend one or the other opinion? You feel free to say if you were also random guessing, because I guess there's a chance out of 1 out of 2, so it's not a bad choice.
I was like guessing because you shouldn't ask, you wouldn't ask me if the answer wasn't like strange. So strange that bacteria have.
So you guessed against your intuition.
Yeah.
Interesting. Yes?
Same, but maybe bacteria are like less specific and they have to do everything like as one, let's say, cell.
Well, yeah, so they need to have more complex features because they can't rely like on organs and stuff like that. Okay, good reasoning. What about the other camp?
But you said it was the opposite as for, you said it's not like, I'm not talking about the genes, and I think it's more complex than the genes for the
Yeah.
compounds or whatever. Is that the human?
Yeah, so that, I don't know, okay, so wait, I'll come to that. So the answer is eukaryotic proteins, in particular mammalian proteins, are more complex than bacterial ones, so good job to those who guessed that, but it doesn't matter because I'll come to why that is in a second. What did you just say? Oh, the number of genes. So this is really complicated, and I think we don't understand why very well. Bacteria have a low number of genes, in fact. Humans have an intermediate number of genes, and there's an organism that I think you started talking about plants. So I'm fascinated by the fact that the Christmas tree is like super complex. It has so many genes, and you know, if you compare a human and a Christmas tree, you know, you might argue that a human is more involved, but the number of genes doesn't correlate with complexity at all. Okay, about the proteins themselves, it turns out that the complexity of the proteins in bacterial organisms and in eukaryotic organisms are actually quite similar. So they're not more complex in one or the other, except for the fact that eukaryotic organisms can undergo what is called post-translational modification. Have you ever heard this? No? Okay, so this is, we usually tell you about DNA, RNA, protein, but we ignore this last step, which is post-translational modification, in which there are enzymes that will come and modify the protein. So this is glycosylation. You might have heard phosphorylation, this kind of words. Those mean that the proteins get modified later. glycosylation means there's going to be sugars that come and decorate the proteins and modify their function. It's the same for phosphorylation. So eukaryotic proteins are more complex, usually, than bacterial ones, not because they have more domains and so on, but because they have the ability to be post-translationally modified. So it's all subtle, I don't know. I think sometimes it's difficult to explain why we can just observe and like try to explain through physics and evolution why this might have happened, but anyways, this is how it is. It's only possible in eukaryotes, the opposite. So bacteria don't have the enzymes that allow them to post-translationally modify. Yes. So coming back to this question, the people who were on the team that eukaryotic proteins are more complex, why do you think bacteria do not want the complex proteins? It's about cost. It's about energetic cost. Bacteria are under a lot of pressure, so they want as simple a life as possible. So for them, putting in glycosylation and so on, that would cost too much, and it would not confer them an evolutionary advantage that would be as good as losing the energy. Okay. So just, you might notice we can always, I think, we can always make arguments. You need to be a little, clever, because it's, as you could see, it's not so difficult to defend one or the other argument because biology is so complicated. And regulation can happen at so many levels of gene and expression and number of proteins and switching on and off genes and so on. So just get used to kind of arguing one or the other based on all the knowledge that you've accumulated. Example taken from membrane proteins. Do you guys remember this example? This was a potassium selective ion channel. We kind of saw it from the side. Remember, it was these two helices, and then there was the selectivity filter where the ions were going through? Yeah, maybe? It's pretty cool. It's what I do research on, so you should be fascinated. So these are the two helices I was talking about. We're looking at it from the intracellular domain, so the ions would be flowing through in that direction. And this, there are 4 subunits that are identical. And this equivalent protein in eukaryotic compared to bacterial has the same architecture here in the middle. You can recognize it. It's oriented the same way, but it has extra domains. 4 of them, still a tetramer. Remember, this was what was called a voltage sensor domain, and it was able to respond to changes in the membrane potential. And through changes in these domains, it was changing how the pore was opening and closing. So bacterial channel, these 2 objects have the function of conducting potassium. Bacteria have this channel, which is gated by pH, whereas eukaryotes have these auxiliary domains that make them gated by voltage. And you could argue that eukaryotes need to transport the electrical activity along their nerves and their muscles, so they need to be able to respond to changes in membrane potential, whereas bacteria need to be able to respond to changes in pH, because this is something that bacteria will be exposed to, right? They won't really be exposed to electric fields. Traditionally, they'll be exposed to pH changes. So an illustration of conserved structures that then get decorated in eukaryotes. Very quick story. I think traditionally, if you're asked what's the timescale of evolution, how long does evolution take, I think we think of millions of years, right? tens of millions, hundreds of millions. And this is an example that shows that evolution can occur actually much faster than this. So actually another example is probably bacterial resistance. I guess you guys have all heard about this and how by using too many antibiotics, the proteins are evolving and developing new strategies so that they can escape the antibiotics. So we need to always come up with new antibiotics, and it's a major problem in the world. So that's one way to know that these evolution occurs over our lifetime, right? It's not over, not necessarily over millions of years. This was an example of a study that was done on the cod, which is a fish that lives in the Hudson River. And they looked at specific enzymes that make them resistant to this pollutant called PCB. And they basically correlated the sequence and the structure of this enzyme with the concentration of PCB. And they could see that there was a direct relationship between the quantity of PCB that you have and the proteins. And we started using PCBs not that long ago, and then we stopped also. So with this kind of study, you can really look at how evolution is operating on the past 50 to 100 years rather than over millions of years. Okay, so now that we've done all this introduction and I've quizzed you on many things, I hope you have an idea of the types of questions that we like to ask. We're going to tackle our big question, and we're going to start looking at how nature stumbles always on the same patterns from very different sequences. And to do that, we will have to consider these specific points. So now, even though you didn't want to answer my question earlier, you know that the main driver for folding is hydrogen bonding. Actually, you did answer my question, I remember now. So it's hydrogen bonding, which means that loops and coils cannot be inside of the protein because we don't want the backbone carbonyl groups and NH groups to be exposed to hydrophobic parts. And so what this means is that the edges of helices and sheets, the loops, they must be facing the water, whereas the secondary structure elements need to be placed together. So do you think it's possible, based on what I just said, to have a single beta sheet inside of a helical region? Is that favorable structurally, energetically?
Inside of helix.
So one of these yellow strands with helices around it. No other beta sheet, just one. It's a bit far. Why?
I mean, we always learn that beta sheets are stable because they establish hydrogen bonds with other beta sheets.
Yes, exactly. So if you had one beta sheet only, then it would have the carbonyl groups and the NH groups on one side that would not be paired with anything. And since the helices around them would tend to be hydrophobic in the middle, then this would mean a lot of moieties that would like to form hydrogen bonds that would be exposed to hydrophobic media, so we wouldn't have it. That's right. So the more common form is to have beta sheets that are then embedded into helices, which means that you'll see in all of the folds that we see, usually the helices and the sheet regions have to be separated, and that is mainly because the beta strands need to be together, not because of the alpha helices themselves. And it will be very expensive to have structural defects. And by defects, I mean exposed hydrogen bond forming type of moiety. There are exceptions, but you have to pay some type of energetic price. I think in one of Eric's lectures, you talked about cysteine disulfide bridges formed by two cysteines. So this is when two cysteines, which have an SH group at the end, they come together, and under reductive conditions, they can form a disulfide bond. This gives a lot of energy to the protein because it's a covalent bond formation. And with this, you might be able to then expose some hydrogen bonds. So I'm not saying that you will never see this, right? I'm just saying that it's not common, and it's not common because there is a price that you pay energetically. Another source of energy, remember salt bridges? You know what those are? When a positively charged amino acid and a negatively charged amino acid form an ion pair. Yeah, so these are also good sources of energy because electrostatics is a very strong type of energy, especially when it's in a hydrophobic region. So we can have defects but we need to compensate this with disulfide bonds or salt bridges. So this comes to now thinking of structures in terms of layers. And like I showed you before in this classification where we had this diagram with the red, green, and yellow quarters and halves, we could see that most often these structures are arranged in several layers. And the most common is 2 layers. Why is that? So by layer, I mean either a beta sheet or helices that would be arranged next to one another. If you have only one layer, it's not a very kind of useful fold because it's not a fold precisely. It's like taking one sheet of paper, right? It's like, it's boring. Nothing can really happen on it. But if you have 2 layers now, you can start thinking about interesting biological activity because something can happen in between these 2 things, right? And we talked at the very beginning about burying a binding site or having an ion conductance pathway. For these things to happen, you need to have at least 2 layers. 3 layers do exist. The typical thing is the Rossmann fold, which contains beta sheets in the middle and helices on either side. And in that case, you can have 2 different cavities on each side, right? You could have, so basically with 3 layers, you have a binding, something that can happen at this interface and then something that can happen at this other interface. And then it's interesting that you really never see 4 layers or 5 layers. 5 you never see, 4 is very rare. So why might that be? Do you guys remember what's the proportion of hydrophilic versus hydrophobic residues in nature? (50-50). So we have about 50% of amino acids that are hydrophilic, 50% that are hydrophobic. And for the sake of the argument here, I'm talking only about globular proteins that live in solution in water. So if I have 2 or 3 layers, I can put half of my hydrophobic amino acids inside and half outside. Now if I start having 4 or 5 layers, imagine 5 layers means the 4 layers that are in the middle need to be hydrophobic, and the 3 plus 2 halves need to be hydrophobic, and the outer layer needs to be hydrophilic. So now I'm really off balance. I need to bury much more hydrophobic residues than I have available in nature. So this is a reason that we don't really have 4 or 5 layers. As always, there are exceptions, but mostly we don't. And this is the reason why we mostly have 2 or 3 layers. So what does this mean? Considering what I just said. If I have a very large protein, this exists. If I have a protein that contains 1,000 amino acids or something, then they will need to be divided into subdomains because I will never have something that contains 5 layers. So this means that my large protein will be made of different domains that then get placed together. I showed you hemoglobin. That was 4 globins, for example, 4 globin folds, right? And this is true of many different proteins. So, like I said, what we're asking today is the opposite of what we've been saying before. And it's asking what folds can, instead of asking in which way a sequence folds, we'll ask which sequence can fit a given fold. And this is just a real simple scheme of the type that physicists like. Imagine that we have 2 types of amino acids, hydrophobic and hydrophilic, again. And these are different types just kind of folded on the lattice. Then the most simple folds can accommodate a lot of sequences. And this is why they are common. So basically, this one here, for example, let's say contains 2 amino acids of one type and then, 10 amino acids of the other type. This will be difficult to find, so this will not be very common. Whereas these kind of random ones where you have 1 of each that are alternating, these will be much more common because they can accommodate a lot of sequences. And so specifically, for example, I was mentioning cysteine disulfide bridges before. If we want to put a disulfide bridge, we need to have exactly a cysteine in the 2 sequences that will come together. So this does not tolerate any other amino acid. So this will mean that this kind of fold will be uncommon. And remember how I was telling you that these are the reasons why you can have defects in terms of having exposed hydrogen bond partners. So we don't have a lot of hydrogen bond exposed because we would need to have a cysteine bridge, and a cysteine bridge doesn't happen often because of entropy consideration. Quick reminder, then we take a break, just to see if you've been kind of looking at the early lectures. Eric introduced you to this Greek key motif, which was 4 beta strands that were arranged in this specific fold. And it's always intrigued, I think, biologists that this looks very much like the type of patterns that you see on the Greek vases. And basically, just to make it short, this type of pattern is a way that you can take one line and you can fold it in a way that the line doesn't intersect with itself. And artists really like patterns, and apparently biology also. So that's why you find this kind of arrangement. If you think about, when you go home and you fold your sheets or your, what's it called? Towels. The aim is that it takes less space, right? So you don't kind of just like put them in your cupboard with like one sheet. Instead, you fold it. And this is the type of fold, actually, if you look the cross-section of folding a towel, this is what it looks like. So this is kind of the natural way, basically, that you can fold something. And of course, because the towel is a real object, it can't intersect itself. So, quick reminder of the fact that this is called a Greek key motif. And we're going to take a break, and then I'll continue, and we'll build this model and explain why probabilistically we can only observe these little folds into little domains. So come back at 1:15, and then we'll take the rest of the lecture.